So as I was mentioning, I'm a postdoc with Shan, and I'm going to be explaining a project that I've been working on for several years now. That actually started when I duped Anna into thinking that this was a good idea, and then I transitioned into duping Shan that this was a good idea, and I'm going to dupe all of you into hopefully thinking that this is actually a good idea. So I want to start with with the agenda, right? There's a lot of people new here that don't know me, and there's also a lot of people that already know me from quite a while ago, but still don't know what I do. So hopefully this is gonna answer the question of what does Julian do with his time at the globe? Why is it interesting? This is a very important part, but most importantly, why is it useful and why you should care? So I'm gonna start with this figure that I hope it's been shown in many other presentations, but I'm going to explain it quickly. In the x-axis, we have a year, and in the y-axis, we have a cost of sequencing. And you can see how, as the years have passed, the cost of sequencing has plummeted tremendously. I want to point out that the y-axis is in logarithmic scale. This means that the cost of sequencing has decreased exponentially with the years. More importantly, we have the white line that says there Moore's law. And just a reminder or a recap, Moore's law is just an observation that Gordon Moore, the founder of Intel, a microchip manufacturer, it was an observation that microchips will double the transition count every 2 years. And when you double the transistor count, you increase the computational power. So that white line just means that computational power has also been decreasing exponentially through the years, and computational power has been the underpinning of human development for the last 70 years. So I want to contrast that with sequencing cost decrease, just so that you can see just how much progress has been done. And the consequence of that progress is that we are able to produce massive amounts of data, so much so that data now presents a bottleneck for our analysis. Not only that, having such amounts of data also presents new opportunities of how to analyze and how to think about that data. And that's what I'm going to go with the rest of the presentation. So when thinking about this massive amount of data, what I like to think is what fundamental properties do sequences have, right? And of course that they do have properties, but I'll make that clear in a while, because once I convince you that sequences have some higher abstract fundamental properties, what are those properties? Which is a very difficult question to answer. But lastly, what are the practical implications of trying to figure out what these fundamental properties of sequences can be used for? So for that, I have to first explain the Mexican sombrero of insights, because this is actually, I'm going to be talking about a very computational and mathematical project, and that has the property that if I start to get very technical, the insight that you're going to gain is going to be quite poor. But if I skip all the details, the insight that you're going to gain is also going to be poor. And everybody has a different Mexican sombrero of insight depending on what they're being explained. And I'm really hoping that I'm going to reach the sweet spot for most of you. And even though this is going to be computational heavy and mathematical heavy, I promise that we are not going to be in the right side of the technicality side of things. So for that, let's start with an image of a dog. And I hope that you all agree that this is a dog, and it was very easy to recognize that this is a dog. And some of you might recognize which particular dog this is, right? What I want to point out is that it was super easy to recognize that it was a dog. And nowadays we have algorithms that can also easily recognize that this is an image of a dog, right? What's funny is that if I switch things around, we can no longer recognize that this is a dog. I haven't changed a single pixel value of this image. I just scrambled them around, and now there's no way of recognizing that this is a dog. And why is that? It's because this image of a dog, or dogs, have fundamental properties that give them their dogness, right? When you're looking at this image, you don't go into your database of all the dogs that you have seen before to match them and see like, oh yeah, it matches all these dogs, and thus this has to be a dog. You just instinctively know, human brains can instinctively know, can recognize properties of an image in order to classify them for whatever they are. What's really impressive is that I can change the setting completely, and you still know that this is a dog, right? There's some invariance to this property that when I switch the image around, depending on how I mix it up, right? If I mix it up like this, there's no dog. If I mix it up like that, that's still a dog, right? Doesn't matter how ridiculous the dog looks. What's even more curious is that when I show you not a real dog, you still know that this is a dog, right? The fundamental properties of dogness are still there, even though this is not a real image. So that leads me to the next question. What are the fundamental properties of this sequence such that we could tell that it's a Resolvium sequence, right? Why can we do that with images that can have as much data as a sequence? But with sequences, we have to go to all these complex algorithms in which you do alignment, we do complex mathematical tasks. We have to recompile, again, these huge databases just to maybe find out that this actually was a sequence pulled from a Resolvium bacteria, right? So once again, what are the fundamental properties of a sequence that we can use for things like assigning it taxonomy, or maybe a gene content, or maybe evolutionary relationships? How can we, what I'm working with is create a model that can extract these properties that I'm calling fundamental, that give this thing its Resolviumness and any other sequence its own, tagness. So how I'm going to go about this, I'm going to present the model. I'm going to present how I'm implementing that model, and then I'm going to present some curious things that I have been able to do with this approach. Okay, so in broad strokes, the mathematical model that I'm using to do this property extraction is called an autoencoder. Okay, and this is all the math that you're going to see today, and it's actually pretty easy because an autoencoder has two parts. It has one first function, the one on the top, that takes some data, that's X, and will encode it, that's why it's called FE, into some code Z. Sometimes this code, I'm going to call it an embedding. And a lot of the times, the dimension of that code is smaller than the dimension of the input data. Okay, so that's the encoding part of the autoencoder. Now, because there's an encoding part, there's also a decoding part. And that's the second function here, that takes your embedding, and then from the embedding replicates the original input. Okay, so another way of viewing this mathematical model is with a neural network approach, in which you have your input that are these first gray balls. The lines just represent mathematical operations, and then the blue balls represent the code or the embedding that is of a lower dimension than the input data. And then again, there's mathematical operations that are the decoding part that just try to replicate the original input as the output. Okay, and because the code has a lower dimension than the original input, the autoencoder is forced to extract what is really informative of whatever input you're giving to it. Okay, so what I've been doing is using autoencoders to grab the massive amounts of sequencing data that are available to try to generate these models that can extract properties from sequences by doing this operation. So the implementation of this is that first, sequences are going to be vectorized. Well, sequences are going to be seen as vectors, n-dimensional vectors. And I'm going to explain this in the next slide. But the idea is that from a sequence, you can convert it into a point in high-dimensional space. Then I'm going to use an autoencoder implemented via a neural network. And finally, I'm going to show you some applications of doing that for classifying sequences taxonomically. So let's go to what I mean by sequences as n-dimensional points. So there's a concept in sequence analysis called the Kmer, in which for any given sequence, you can extract a subsequence of a given length. In this case, it's of length 7. Okay, and when you extract those subsequences, you can count them, and then you have what's called a Kmer spectrum. That is just how many times you see each Kmer. One problem with this approach is that if your Kmer is really large, then the possible number of Kmers that you can obtain is just impossible to compute efficiently. So I'm using this different approach in which for any given Kmer, you only take into account certain positions. The positions are the ones that you find a 1 in this binary vector. So that means that any given Kmer becomes a shorter Kmer, and 2 distinct Kmers can be counted as the same Kmer as long as they coincide in these positions. So there's some loss of information, but at the same time, the number of Kmers that you can track becomes more traceable. And in the end, what ends up happening is that any sequence can be represented just as a vector of counts. And a vector is the same as saying a point in high dimensional space. Okay. All right. So how do I train this autoencoder? So just as a recap, we have sequences that I convert into high dimensional points. And then I'm going to use this autoencoder as a neural network to train the autoencoder to replicate the input as the output by first transitioning into this embedding that is of smaller dimension. And for the result that I'm going to show, I'm transforming sequences into vectors of 1024 dimensions. And then the autoencoder reduces those 1024 dimensions into 64 dimensions only. Okay. So what we're seeing here is the actual training process of the autoencoder. And just I'm using RefSeq data. RefSeq is just the reference sequence collection from NCBI. It's around 2.7 terabases of sequence, and I'm throwing everything at the autoencoder: eukaryotes, viruses, bacteria, archaea, the entire tree of life. Okay. So I want you to focus on the graph here, and the x-axis, the one that says batch, just means how many times have I gone through the entire training data set. Okay, and the y-axis is how different the output is from the inputs. That's and it's in the neural network or machine learning lingo, that's called a cost. So if the output is very different from the input, the autoencoder is failing and the cost is really high. And that's why we see that at the beginning of the training process, the cost is high. But as we repeat the process of training, the cost starts reducing even progressively, right? But you can see that there's 2 lines, the red line and the blue line. The blue line is the cost of the training data set. That means the sequences that I'm using to actually tune the model to do what I'm telling it to do. And the red line is a disjoint data set that the model has never seen. So that because training a model only on the data that applying a model only with data that you're training it with doesn't make a lot of sense, right? You want it to generalize into data sets that it has never seen. So we can see how the model, I mean, in the validation data set, so the data set that it has never seen, it's still learning, but it's the cost is higher than the training data set. And this is just it's a classical example of a slight overfitting, in which the model works really well with the data that I use for training, and not as well, even though it's still learning with data sets that it has never seen. And that always happens, especially with these models that have hundreds of thousands of parameters. Okay, so just quickly, one of the concerns that I had with this is that, well, maybe the model is doing what I'm telling it to do about replicating the output, but maybe in the embedding is doing something completely different. It's taking sequences that should be similar to each other and then just putting them far apart in this lower dimensional representation. So to see that the autoencoder actually does something useful with the sequences, what we have here, each point is a pairwise distance between 2 random samples of sequences. And what we have in the x-axis is the distance between those 2 sequences in the large dimensional space. And then the y-axis is their distance in that embedding. And what I'm trying to show here is that 2 sequences that are close together in the vector space are going to be together in the embedding space. And as the distance increases in the vector space, it will also increase in the embedded space, okay? Which hints at the usefulness of using this embedded, this reduced space for doing something with the sequence, right? But what is happening with the sequences? Okay, so I grabbed from RedSec again, I sampled 1,000 sequences from 4 bacterial genera, Burkholderia, Escherichia, Pseudomonas, and Yersinia. I encoded them with a trained autoencoder, and then this is a PCA of that of those sequences when they have been transformed into the embedded space, into the low dimensional space. And you can see how the structure, the taxonomic structure of the sequences is conserved, right? We have our Burkholderia, we have our Pseudomonas, we have the Yersinia, and we have the Escherichia. What's also really cool is that, for example, Yersinia and Escherichia that are clumped together in the PCA, they're both Enterobacteriaceae. They're both bacteria that are associated with the gut. And then we have what's also really cool is that the second principal component separates Burkholderia, and these Yersinia, Pseudomonas, and Escherichia, they're all gamma protobacteria, and Burkholderia is beta protobacteria, which I argue is a point to say that this model is capable of extracting these fundamental properties of sequences, because when I show this representation, certain taxonomic relationships are still maintained. This is even stronger when instead of PCA, I use these nonlinear manifold projections. So again, we have Yersinia and Escherichia, Pseudomonas, the gamma protobacteria, and then Burkholderia. Of course, something happens with sequences that suddenly don't match with their group. And I know that some of them happen to be sequences where there's like viruses that were inserted or repeat regions that are very similar in all groups, and that way we could place something in the incorrect group. But that's still a hypothesis. Interestingly though, is that so the model I threw in bacterial data, and now I'm throwing it data from a completely different domain in life. This is Archaea. This is at the class level, and the taxonomic relationships are still maintained. Which again, I argue this has to do with this autoencoding approach that actually can extract, the fundamental properties of these sequences, such that the evolutionary relationships still make sense. And lastly, I just want to show another practical application that I've been working now that Ignacy has joined the group. Ignacy is a master's student that has been helping me with model metaparameter exploration. And together we are developing this taxonomic classifier. So the idea is that you have a sequence, and then you can quickly assign it a taxonomy based on this autoencoding approach. So what we do is that we grab data, we encode it with our autoencoder, and then we throw in another neural network that will tell us what label that sequence belongs to. And what I did to test this first is that, we grabbed 100,000 bacterial sequences from RedSec, which represent around 7 gigabases of sequence, and the minimum length of one kilobase. And then we classify them in 12 groups. There are the 11 most frequent phyla plus the rest. Because what happens with RedSec is that not all phyla are equally represented. There is a bunch of protobacteria, but there is very little of other phyla that are of less interest. And again, this classifier was, this data set was split 70% for training, 30% for validating that training. And if you focus again your attention to the plots, we have the training cost and how it starts reducing at each batch, but the validation suddenly plateaus. And that's where we started seeing just pure overfitting of the model. What is cool is that so far with this first approach, we are obtaining 87% accuracy rate, which means that we're getting wrong 1 every 10, like 1.3 sequences per every 10 sequences, which is still not great. I believe that we can take that way further. But the good thing is that the amount of sequences that we can process is just massive. Again, all these programs have not been optimized, but so far we are able to classify around 3,000 sequences per second. And I think that we can get like much higher. the cool part of this as well is that here we're doing taxonomic classification, but the label can be anything. Like you can grab a sequence and just the label can be, is this gene present or not? Or is this combination of genes present or not? Or is this virus present or not? And that's where we're heading to next, in which we want to do some model refinements, which means that we want to, right now we're using very simple neural networks that are not very big. But fortunately now with the new server that has some really nice GPUs, we can take these very simple models that we were using, and it's several orders of magnitude that we can increase the number of parameters. Data expansion. So right now we're only using RepSec, but there's so many other sources of sequence. We're thinking about more applications. So for example, with the taxonomic classification, one thing we want to do is, for example, to detect horizontal gene transfer. If you grab a genome, you start scanning it, and as you scan it, you should get the same taxon all the time until maybe you reach a region that if it was prone to lateral gene transfer, then you're going to get a different taxon, and do that on a massive scale across the tree of life. There's a problem of the black box problem, which means that apparently the models are working as I have hypothesized, but I don't know what the model is doing. I don't know exactly what these fundamental properties that the model is extracting, what they are, what Kmers are they, what sequences are they. And finally, the toughest of all of this is to make all of these models and applications usable by other users, which is quite hard. But that would be all for me.